When we look at the white and gray matter in the brain and the spinal cord, we need to understand what they are, right? What do they contain? So gray matter contains short, non-myelinated neurons, as well as the neuron cell bodies. And it also typically is more of a central cavity surrounded by gray matter. We can see that in our spinal cord here, right? So spinal cord here has this central cavity surrounded by gray matter. The brain will see that in the brain specifically, the outer component is consisting of gray matter. White matter is both myelinated and non-myelinated axons, really no neuron cell bodies here in white matter. And white matter in the spinal cord is external to gray matter. In the brain, it is internal. I mentioned ventricles. Ventricles are fluid-filled chambers that are continuous to one another. They are all interconnected, and they're filled with cerebrospinal fluid and lined by ependymal cells, which are a neuroglial cell type. We start with our paired lateral ventricles. These are large, these are C-shaped in size, and they're located deep in each hemisphere. They are paired. The pair is separated by this membranous septum pellucidum. That is the separation between the two paired lateral ventricles. From there, the lateral ventricle is connected via the interventricular foramen. The interventricular foramen is what connects the lateral ventricle to the third ventricle, and the third ventricle lies in the diencephalon. The diencephalon then is connected to the fourth ventricle down below via the cerebral aqueduct, and the cerebral aqueduct is located here. And then the fourth ventricle, located kind of in this brainstem region below in the hindbrain, will be kind of become continuous with the spinal cord, the central canal. There are three openings that connect the fourth ventricle specifically to the subarachnoid space. There are lateral apertures that are on either side, and then there's a median aperture, which is located kind of on the roof. And so all these three structures are what connects the fourth ventricle to the subarachnoid space, allowing for cerebral spinal fluid to bathe the brain. The cerebral hemisphere does have specific surface markings. There's gyri, which are ridges, and then there's sulci, which are the grooves. There are fissures, which are essentially deep grooves within the brain. Excuse me. Longitudinal fissure, as well as a transverse cerebral fissure. The longitudinal fissure is what separates our right and left hemispheres, and the transverse cerebral fissure separates the cerebrum and cerebellum, which we cannot see on this image. But we could see it here. This is our transverse cerebral fissure. There are several different sulci that divide the hemisphere into our five lobes. We have our frontal lobe up here. We have our parietal lobes, both a right and a left. We have a temporal lobe, a right and a left, and we have a singular occipital lobe. There also is an insula lobe that's located kind of deep into the brain. You can't see it in this image. The first four are named after their overlying skull bones, so you can associate them with the frontal bone, parietal bones, temporal bones, and occipital bone. The insular lobe, like I mentioned, is buried deep to the temporal, parietal, and frontal lobe. If we look at the cerebrum, it's the largest of the brain regions, and it's divided into two hemispheres. Each can be further divided into the lobes, as mentioned. If we're thinking of the functions of these lobes, our frontal region is in charge of motor activity, behavior, speech, critical thinking, and problem solving. Our parietal is focusing on somatosensory function. Temporal is hearing, smell, and memory. Occipital vision, and the insula is more associated with taste, emotion, and viscerosensory information. There is a central sulcus, and the central sulcus is important because it separates two key structures when it comes to motor and somatosensory function. The precentral gyrus in front of the frontal lobe, that's here, and the postcentral gyrus of the parietal lobe, which is behind. There is also a parieto-occipital sulcus that separates the occipital lobe and the parietal lobe, and that's right here. The lateral sulcus outlines our temporal lobes. Okay, lateral sulcus there. When we look at the hemispheres, they all have three basic regions. There is the cerebral cortex, which we can see kind of on the surface. This is the cerebral cortex. This is gray matter. Beneath the cerebral cortex, there is white matter. And within the white matter, there are basal nuclei. And remember, in the nervous system, nuclei is just a cluster of neuron cell bodies. And so in our deep inner white matter, inside our brain, we have these different areas that are gray matter containing, and they are our cell bodies, neuron cell bodies. buddies. There we go. Harder to draw when we're doing this. There we go. If we are going to try to think physiologically about the cerebral cortex, we can look in their frontal lobe and we can see that there's kind of several key structures. Right in front of that central sulcus, we have our motor region that's going to help us actually communicate to our somatosensory nervous or somatic nervous system what motor functions we want to do. The front, we have more planning activities, planning movements and thought. And then we have Broca's area, which is specifically named for word formation. In our temporal lobe, we have an auditory region and a behavior, emotions, and motivation region. And then we have that's kind of also close to our limbic association area, which makes sense. The limbic system deals with mood. And in that kind of temporal to parietal region, we have Wernicke's area, which is specially named for language comprehension and intelligence. In the parietal lobe, we have our somatosensory region and our spatial coordination and kind of our body and surroundings region that helps us both understand sensations that are coming into the brain as well as then know kind of where we are in space. And then back towards the back of the temporal lobe, we have kind of a naming of objects, which then moves to our occipital region where we have visual processing of words, how we read, and vision. These are our different cerebral cortex regions. Again, you're not going to have to look at a picture and identify based off of a picture, but you should have an idea of what these named areas, what their physiologic functions are most likely associated with. That would be high yield for this exam. When we come back, we're going to get into the primary somatic motor cortex and our somatosensory cortex.